we will be focusing on RNA processing. Some parts of the RNA processing we had already done them. For example, we are done with capping and the polyadenylation. We are done with them in the previous video. In this video, we will be mainly focusing on RNA splicing as well as RNA editing. So here on the screen, as you see, this is the you can say this is a casual representation of the central dogma. We know that the DNA it performs the process known as the transcription, which is one of your fundamental processes, and it makes a pre-mRNA. It doesn't make an RNA, or you can say an mRNA. It first forms a pre-mRNA. You can also call it an immature mRNA. Now, why do we call it a pre-mRNA? Now, there is a reason behind that. The eukaryotic gene, if I'm talking about, especially in the case of the eukaryotes, the eukaryotic gene, they contain two different type of sequences. One sequence we call it the coding sequence, and the one sequence we call it the non-coding sequence. Now, the non-coding sequence is known as an intron. On the other hand, the coding sequence is known as an exon. There are some other definitions that you can also use to define what is an intron and what is an exon. For example, you can also say that exons are the regions of the gene retained in a mature RNA. As you see here, this is a mature RNA. This one only contains the exons, and as you can see, it is properly kept, and there is a polyadenylation tail here. Okay, so exons are basically, you can say, these are the coding sequences present in a eukaryotic gene. Now, basically, the coding sequences are the one that we are going to use, because the mRNA, in the next step that we call translation, the mRNA, the mature mRNA, is going to go out of the nucleus and it's going to make a protein. And that protein, only the coding sequences play a part. So, how do we get rid of non-coding sequences? And here comes what we call splicing. And as the name suggests, splicing just means that we are going to cut and remove the introns or the non-coding sequences. From here on forward, I will be calling the non-coding sequences introns. Okay. Now, once the genetic information is transcripted into RNA, the introns must be removed in order for the RNA to produce, you can say, a working protein. However, there are still some regions of the mature mRNA which are non-coding, but they are still retained. Such exons are known as non-coding exons. I know this doesn't qualify as the, you can say, definition coding, right? So there are some non-coding exons, right? However, I will not be diving deep in that topic. For example, let me tell you, in the mature mRNA, there are some 5' and 3' untranslated regions, also known as the UTRs. Okay, so basically they are exons, right? But they don't code. So that's why we call them the non-coding UTRs, untranslated regions, and they are basically present, as mentioned here, on the 5' end as well as on the 3' end of the mature mRNA. So the mature mRNA still contains some non-coding sequences, but they are still exons. Because again, the technical definition of an exon is that it can be in any region of a gene which is retained in the mature mRNA. On the other hand, let's for example, some RNAs also contain, for example, let's take, there is RNA known as the cyst regulator. I hope you have heard of it. If you have not heard of it, we will be discussing about this in a further video. Okay, and this is basically the cyst regulator. It is a non-coding RNA. Now, before moving any, let's move forward. And as you can see, the eukaryotic gene, as I mentioned earlier, it contains both, you can say, the coding sequences, the exons, and the non-coding sequences, exons and introns. Now, as you can see, this is our DNA, the genomic DNA. The green part that you see in this region is the exon, and you can say the segment which is present between two exons. This is exon 1 and this is exon 2. The segment present between two exons is an intron. So here we have 4 exons. So technically there will be 3 introns. We name them intron 1, 2, and 3. So first of all, there will be transcription. So this DNA, it undergoes transcription using this promoter element that it has, right? The RNA polymerase will attach on the promoter element with the help of some transcriptional factors, and it will produce what we call a pre-mRNA. Now, this region, right? You see these two regions. These are the one that I was talking about earlier. This is the 5' UTR and the 3' UTR. UTR stands for untranslated regions. So as you can see, they are still present in the mature mRNA. This is the mature mRNA, or you can say a spliced mRNA, because it is not matured yet. And a matured mRNA, it contains a 5' methyl cap, guanine methyl cap, obviously, or you can say 5' methyl guanine cap, a poly A tail, right, and only the exons, right? So, however, it still contains some untranslated regions, as I have already shown you here. Now, as you can see, this is what we call a pre-mRNA. You can also call it immature mRNA. And what we are doing here, we are gonna only keep these guys, which are the exons, right? And we're gonna remove what we call the introns. Now, if I'm let's move forward, and before moving forward, I would like to talk a little bit about how much exons are there. You can say the number of exons and the number of introns, the size of exons and the size of introns. So basically, the number of introns in a gene, it can vary. It can vary, for example, in the case of, let's talk about in the case of yeast, right? Let's take Saccharomyces cerevisiae. The genes of the Saccharomyces cerevisiae usually contains one or two, you can say, introns in most of its gene, right? So let's take just one in the case of yeast. It becomes, for example, let's move to a higher organism. Let's take a chicken, right? A chicken can contain up to 50. For example, I'm here talking about a particular gene in the case of the chicken. There is a gene known as pro alpha 2 collagen gene. It contains around 50. Now, let's move to the most complex organism present on this planet, humans. The humans, they can contain, you can say, up to, I think, let's talk, there is, I think, the titin gene, as I think it is mentioned here. It can contain up to 363 introns in one gene. So, again, as you can see, uh, the number is variable, but usually what is the case, higher the complexity of an organism, higher will be the number of introns. It also depends on the number of, basically, on the complexity of the protein, right? So it varies. Now, size of the, let's talk about the size of the exons and the introns. Now, as you can see here, basically, and as you can see here is a graph where on the one hand, I have put the average number of introns per gene, and here are the organisms, right? So here, this is the Leishmania major, which is, as you know, it is a protozoan. And then here comes the yeast, the Saccharomyces cerevisiae. There is this Drosophila melanogaster, and then there is C. elegans, a nematode, Thaliana, Rhabdospicus thaliana, right? Mouse and humans. So as you can see, there is a certain trend to this graph. These are, you can say, the Leishmania that we have, Trypanosoma is also here. They doesn't contain, the average number is almost equal to 0, right? So, but as the complexity of the organism is going up, you can see a definite trend here that the number of, average number of the introns per gene is increasing. And that it goes up to the humans, which contain around 7.5, which is the average number of introns per gene, right? Because again, you see, mouse and humans both are mammals, which are technically speaking, are the, you can say, the most complex organism present on this planet Earth. So, using this, I would like to say that you can say that the number of introns per gene increases with the complexity of the organism, right? As it can be observed from the graph below. Now, let's talk about the basically the size of the intron and the exon, right? Before moving forward, next we will be discussing the chemistry of splicing. But before that, let's talk about the size of the introns and exons. How large are, you can say, introns and how large are exons? Here, I'm going to take an example. For example, there is an enzyme. I don't think you have heard of it, or maybe you have. The name of the enzyme is dihydrofolate reductase. So, dihydrofolate reductase. So this is our enzyme, right? Now first we are going to see the size of the gene, right? This is a protein and the gene size here is around, you can say 31 kilobase pairs, KB I'm going to write, long. Now, it contains this dihydrofolate reductase, it contains around 6 exons, right? So out of this, there are 6 exons and the remaining obviously will be your introns, right? If there are 6 exons, how many introns should be there? That's a question that I have to ask you, right? So you have to comment how many number of introns are there in this gene if there are 6 exons, right? And again, I'm going to repeat the definition of introns. Introns are basically the segments of the gene, or you can say the DNA present between two exons. So how many introns are there if there are 6 exons. Now, the 6 exons in total constitute about 2 kb, right? 2 kilo base pair, basically meaning 2000 base pairs, and the remaining, which is around 29 kb or 29,000 base pairs, they are introns. So from here, you can get an idea, right? I'm going to give you a rough number. For example, around the exons, they take around 10% of a gene. So 10% of the gene is taken up by the exon. The remaining 90% of the gene is taken up by the introns, right? And their number we have already discussed. Now, thing, before moving again any forward to the chemistry of the splicing and all that, what are the benefits of having introns? That is the next question that pops up in our head, right? Basically, out of this 31 kilo base pair or 31,000 base pairs that we have, only 2,000 base pairs are making the actual protein. Then what is the role of this 29,000 base pair introns, right? So basically, for example, let's take the example of the prokaryotes. The prokaryotes, they don't have introns. Their genes are uninterrupted. So in the case of prokaryotes, we can simply say that there are no introns, right? Because their genes are uninterrupted. But in the case of eukaryotes, and as we move up to the complexity ladder, the you can say the introns and exons are present, and their number significantly increases. Right now, the main benefit of having the introns is you can from the same transcript, or you can say this is what we call a transcript. You see this thing, the pre-mRNA. You can also call it a transcript, right? From the same transcript, you can get different proteins, right? There is a process, right? We call this exon shuffling, exon skipping. There are a number of different type of processes that can allow that. We will be discussing that later in this video. Now, right? So that is the first thing here. For example, in order to get a better idea, let's take another one, right? Let's take the human dystrophin gene, the largest one, right? Now, the pre-mRNA, let's talk about here, about the dystrophin gene that we have. If you don't remember the dystrophin gene, this is related to DMD, the Duchenne's muscular dystrophy, and the pre-mRNA, basically, which is the main, you can say, transcript. Let's just write pre-mRNA to keep it simple. It contains around 24 kb, right? 24 kb. K means kilo and B means base pairs. So basically 24,000 base pairs. So now I hope you know what is the speed of transcription. The speed of the transcription is, let's take an average actually. The speed of transcription. The speed of transcription is around 40 nucleotides per second. So as you can see, it is not that fast, right? As compared to the, you can say, the process of DNA replication, it is not that fast, right? So the first question that I have to ask you is that how much time is it going to take for making one pre-mRNA of the dystrophin gene if the speed of transcription is 40 nucleotides per second, right? If you know the answer, if you can pause the video and calculate the answer. After finding the answer, you can comment it in the comments, obviously, right? So it will take, I have already calculated it, around 17 hours. So as you can see, in order to make just one transcript of this dystrophin gene, it takes 17 hours, right? Now, the primary transcript, which obviously contains the introns, the pre-mRNA, it must be removed. And the process of removing those introns is what we call the RNA splicing. So, the introns will be removed and then it will convert into a mature mRNA. Okay. There are also with particular genes, there are splicing patterns that we will also be discussing. There is something that we call alternate splicing, exon shuffling, actually exon skipping can also result in that, but it is error in splicing actually, exon skipping, so leave that. But alternate splicing can happen, right? And from one transcript, basically from this one pre-mRNA here, you can make multiple proteins, and all those proteins will be isoforms, right? So from this one, you can form isoform. From you can say by alternate splicing, you can make multiple proteins, and those proteins will be referred as isoforms. If you don't know what is an isoform, I don't blame you. Isoform is basically, you can say, isoforms are closely related proteins that basically you can say that originate from a closely from a single gene, or you can say a closely related gene family. So those are what we call isoforms. So that is, I think that is enough introduction. Let's move to the main thing, the chemistry of the splicing, right? So the chemistry of the splicing, as you can see here, there are first thing that we need to discuss is what we call the splice sites. And splice basically means making a cut, right? So there are three main sites that we need to discuss here. Those are, we have the 5' splice site. As the name suggests, it is present on the 5' end of an intron. Just remember this thing, right? Of an intron, not an exon, on an intron. For example, here we have an intron, as you can see, and this will be the 5' end of the intron. So this is our first site that we call the 5' splice site. Then the 3' splice site, which is present on the 3' end of an intron. So this is known as the donor site, okay, and the 3' site, this is known as the acceptor site. Now in the middle there is also known as a branch point site, which usually this is the branch point site where there is usually present an A. However, in some cases it can also be a G, a guanine. Here it is an adenine, right? So this is your branch point. So let's write it down. And as you can see, there is also, it is the branch point is it is followed by a polypyrimidine tract, or also known as the PI tract. Okay, so it is usually present in this area. It is not shown in this picture, but I'm going to mention it anyways. So this is your PY tract, and this is basically your you can say the sites. There are three sites and a tract, PY tract, the polypyrimidine tract. Now there are some conserved sequences here. You can also see them on the screen. For example, the 5' splice site has a GU sequence which is conserved. It means it will be found on almost all the 5' splice sites. And same goes for the 3' splice site. They also have a sequence that we call the AG sequence, which is conserved. The branch point site, as you can see here, this is an A nucleotide, an adenine, which is present in the middle. However, in some cases, as we will be discussing forward, it can also be a guanine. Okay, so bear with me. Now, these are, you can say, the conserved sites that we have. As you know, the splicing involves the removal of an intron in the form of a lariat. Now, you can ask me, what is a lariat? It is a simple thing. This kind of a structure, we call that a lariat, right? And in order to do that, you have to do a certain reactions. So as you can see, this is the chemistry of splicing, how it works. So let's get a, you can say, a brief picture here. So the first, during the splicing, there is a formation. The intron exits in the form of a lariat, right? This kind of structure, as you can see here, this is known as a lariat, an intron lariat. So let's take how it happens. So it happens in two phases. Basically, two reactions are required here. And the two reactions are trans-esterification reactions, as I have mentioned them here. So you will need two trans-esterification reactions in order to do that. Let's take about the first reaction here. The first reaction that we have, as you can see, it is initiated by this 2' hydroxyl group of the branch point adenine, right? This 2' hydroxyl group of the conserved A at the branch site, it will act as a nucleophile. So it will be acting as a nucleophile, right? And it will be a nucleophilic attack. I hope you are up with your chemistry. So it will be a nucleophilic attack on where on the conserved site of the 5' splicing site, right? This GU, I hope you remember, this is a conserved sequence, GU, and on the basically in the phosphate bond, right? This one, just a second, let me remove this one. This phosphate group will be attacked. It will be a nucleophilic attack, right? And basically what will happen, this bond is going to break. So let's do that. So this bond will break, and the bond will be reformed between the A and the G. Okay, and this is a this thing that forms, right? If you see it, this looks like a T point, or also known as a three-way junction, where basically three paths are meeting, right? So this is also known as your three-way junction. Okay, now this is your outcome, the formation of the three-way junction. Now the second reaction comes into the play. As you can see here, after this bond breaks, there is a free 3' hydroxyl group here, right? And as you know, the hydroxyl, the OH, it is acting as a nucleophile. The oxygen acts as a nucleophile, and it will again, there will be the second trans-esterification reaction, and it will be again a nucleophilic attack, right? So the nucleophilic attack, and where on this conserved sequence of the 3' splice site, AG, this phosphate will be targeted, and as you can say, now these two, this is the, let's say this is exon 1, and let's say this is exon 2. These are combined, and this is what we call, you can say the splicing is done. This thing in the middle, the intron, it will be removed, because as you know, this time this bond will be broken, right? So this bond breaks and the intron is released in the form of a lariat. So this is basically your, you can say the chemistry. However, there is a formation of three-way junction. So let's take a close look. So as you can see, this is your three-way junction. Now, basically, as you can see, I hope you can see, just there are three things. For example, this is your branch point, right, AG, and this is your 3' end, and this is a guanine, GU, from the 5' splicing site. They are joined here with an unusual bond, as you can see, right? And basically, this is how the three-way junction is formed, right? So this is your closer look. Now let's talk about how the splicing is done. This was the chemistry of splicing. We have talked about it, and now let's talk about what are, you can say, the spliceosome machinery, and what is the role of snRNPs, also known as snRNPs, right? So let's discuss that. Now, before going deep into the snRNPs or the snRNPs, there is something that we need to discuss first. The splicing, the process of splicing, it is done with the help of what we call a spliceosome, right? And now what you're going to be asking me, so what is this? So basically it is a, you can say it is a big complex, just like a ribosome actually. In size, in fact, it is almost similar to a ribosome. And just like a ribosome, it is made up of both protein and the RNA. Approximately in one spliceosome, there are about 150 proteins, and there are some RNAs. There are actually 5 RNAs. So let's write that down. So this is the condition that we have, right? And this is the spliceosome actually, which is going to consume a lot of ATP. Now, let me tell you something that I actually forgot. In this process, as you can see, there is no ATP required. The chemistry of splicing itself doesn't require any kind of ATP. So I'm just going to here in the no ATP required. The chemistry solves itself, right? As you can see, the number of basically, in order, if you want to find out that the energy is required or energy is released, just take a look at the number of bonds broken and the number of bonds formed. Because whenever you can say a bond is broken, the energy is released, right? So remember that. So if there is a bond break, you can say breaking, the energy is released. So I'm just going to use this mark, so energy released. During any bond formation, if there is a new bond forming, the energy is required. So remember that this is a very basic rule here. So now as you can see, if the bond breaks, energy is released. If the bond is forming, energy is required. If you basically take account, the number of bonds breaking and the number of bonds formed is equal. As a result of that, the thermodynamics solves itself, right? So for example, let me give you a look. For example, first of all, we are going to break this bond. Energy will be released. That bond will be used to make this bond. Sorry, so that energy will be used to make this bond, right? Now, this bond is going to be broken in a second, and the energy will be released. Now, this energy is used to make this bond. So technically speaking, the chemistry of splicing itself doesn't require any kind of ATP. However, let's go back to our spliceosome, right? Our splicing machinery that we have. The spliceosome, it requires ATP. It doesn't do, you can say it doesn't work itself. ATP is not required by, you can say, the chemistry of splicing itself, but the spliceosome actually requires a number of several ATP molecules for a single splicing reaction, right? So basically, and again, I told you that the spliceosome that we have here, it is almost similar to a ribosome. If you remember ribosomes, in the ribosomes, many of the, mostly the reactions, actually almost all of them, are done by the RNAs, not the protein. The proteins, they take more of a structural approach. It is the RNAs which are doing the catalytic functions. The same goes here. It is the RNAs which are going to do most of the catalysis. Right? The two trans esterification reactions will be done with the help of these RNAs. Okay. Now let's talk about these RNAs. These are, for example, if a RNA is used in making a ribosome, we call it a ribosomal RNA. These are, on the other hand, different than that. These are known as the snRNAs. The snRNAs stand for small nuclear RNAs. Now, as I have already mentioned here, we have 5 of them, right? And the 5 of them are basically your U1, U2, U4, U5, and U6. These are the normal one. However, there are a lot of other snRNAs. Basically, the snRNAs, the small nuclear RNAs, they are divided into basically two classes, right? There are two classes here. The one is the SM class and the second is the LSM class. Now, the SM class is made up of, as I have mentioned here, just remember this, U1, U2, U4, U4 ATAC, U5, U7, U11, and U12. Now, you're going to be asking me, where are we going to use this, right? So the U1, U2, U4, U5, and from the LSM class, which is made up of U6 and U680AC, they are all used in splicing. Let me tell you how and where. So there are basically, for example, I hope you know that the ribosome is of two types, right? In the case of prokaryotes, you have a prokaryotic ribosome, which is 70S. And in the case of eukaryotic ribosomes, you have 80S. They are almost the same, but their RNAs are different, right? Now, the same thing goes here. There are two main type of spliceosome. However, they are all present in the eukaryotes because the prokaryotes doesn't have introns, and that's why it doesn't need splicing. So basically, there are two spliceosomes. Let's discuss that. There are two types here that we have. We have the major and the minor. So major, as you can see it by the name, it is the most common one, and that's why we call it the major spliceosome. The other we have is known as the minor spliceosome. The major is made up of U1, U2, U4, U5, and these are U1, U2, U4, and U5 are SM class, and then a U6, which is the LSM class, right? The minor, on the other hand, is made up of U11, U12, U4 ATAC, U5 is common in the both, right? And the U6 ATAC, right? So this is basically your different things, right? Now the RNAs, the small nuclear RNAs, when they combine with the proteins, they make snRNPs. Now what is this snRNP? Quite simple. They are small nuclear ribonucleoproteins, as I have mentioned already here, right? So small, you can say nuclear ribonucleoprotein, snRP, or you can just call them snRPs, right? The snRPs are basically, you can say, these are the main things here. You can pronounce them as snRPs, right? Now, there are numerous proteins within the spliceosome, right? Which are not a part of the snRPs. Remember, not all 150 proteins are present, you can say, are combined with the RNAs. Some of them are combined with the RNA to form this thing. However, there are other proteins which are also present in the spliceosome, which are not connected to the snRNAs. Now, the snRNPs, they basically, you can say, uh, they do three critical roles. Just a second. Let's move up, and you can say we are just going to write them down here in the middle. So basically, the snRNPs, the SNPs that we have, they perform three critical functions. Three critical, basically, you can say, roles that they have, right? The first one, I call it identification. We will take a look at them when we will be discussing how it happens, how splicing is done. The second one is the catalysis. And fourth one, you can just basically, it is a part of catalysis. I just like to, you can say, this is the catalysis for cleavage, and then there is catalysis for joining. So these are the main three roles of, you can say, the snRNPs. And in order to execute all these functions, the SNPs, basically, they use all different type of interactions. For example, they use RNA-RNA interaction, they use RNA-protein interaction, and protein-protein interactions as well. Okay, so here we have now, as I have mentioned, the major and the minor spliceosome. What's the difference between, you can say, them, right? So here it shows the difference in their splicing sites. This is the U2 type, or you can say the major spliceosome, and this is the minor spliceosome. So don't get confused. Let me just mention it quickly. This is major. This is minor. How do I know that? If there is U2, it is major. If it is U12, it is minor, right? So as you can see, most of them are almost the same. However, there are some differences that might be there. For example, the 5' splicing site, it contains a conserved sequence of GU in the case of the major spliceosome. In the case of minor, there are two possible sequences. It can be AU, it can be GU. Both of them are possible. Now, the branch site, it is, as mentioned here, it shows that both of them have a branching point, right? However, in some, you can say in some text, it is also mentioned in some, you can say, latest text, it also says that a minor spliceosome might have a branching point with a guanine in it. Okay, so just keep that in mind. Now, other differences, for example, in the 3' splice site that we have, is we have an AG here in the case of the major spliceosome. In the case of the minor, there are two possible sequences that we have here. It can be AC, it can be AG. So these are, you can say, the differences. Okay. So U2 dependent spliceosome is just another name for the major spliceosome, and U12 dependent spliceosome is just another name for the minor spliceosome. Okay. So let's see if there is anything remaining here. Okay. Now, as you can see, uh, the snRNPs, their main function is basically in the splicing. However, there is one exception, the U7. The U7 snRNP, it is used not in the splicing, but it is used in basically the histone pre-mRNA 3' processing. This process, right? The U1 snRNP, which obviously is a part of the major spliceosome, it has been recently seen that it also has some other functions that we didn't know earlier, right? For example, it protects the messenger RNA or the mRNA, right, from premature cleavage. Okay, it also helps in polyadenylation. It also helps in other processes, as I have mentioned here. Okay, so now let's get moving and let's. Okay, so as you can see here, these are your steps that are required in the splicing. Okay, so let's go through them one by one. The first one, as you can see, is the initial recognition. It is done with the help of the U1 snRNA. It recognizes and binds to the 5' splice site, right, as I have mentioned here. You can just take a pause and take a look here. Okay, obviously, I'm just going to go through them briefly because we are going to discuss them again in the next slide. So as you can see, the first step is the initial recognition. The second is the subsequent recognition. Okay, so the first thing that you have to, the U1 snRNA recognizes, it is the 5' splicing site. The subsequent recognition, it involves the U6 snRNA, which actually replaces the U1 in order to take control of the 5' splice site. There are the third step that we have is the branch site recognition. I hope you remember the branch point in the present in the middle, which is usually an adenine, right? That is recognized by U2. Now the fourth step, there is an interaction between U6. I'm just going to label, you can say just denote the major one. So U2 and U6 snRNA, they interact. Okay, and next thing is obviously the role of U2AF. So U2 basically means U2, obviously, right? And AF means auxiliary factor. The main function of the U2 auxiliary factor is to identify the polypyrimidine tract, right, and the 3' splicing site. Basically, this is the function of the U2AF. However, it also helps in binding of other proteins like branch point binding protein, BBP. We will be discussing them later on. Now, after that, the BBPs, or you can say the branch point binding proteins, will be replaced. Okay. Then there will be the loading of snRNPs. The other snRNPs which are required, they will be loaded on. Then there will be RNA-RNA interaction because the pre-mRNA is an RNA. The snRNA is also an RNA. So they basically interact with each other. Basically, I have mentioned it here, but you don't need to worry. We will be discussing it in detail later on. Now, the release of the spliced mRNA, this is you can say the second last, and the spliceosome disassembly, just like a ribosome, after the translation is done, the ribosome disassembles. The same goes for the spliceosome. Now, let's move to the actual process. So, as you can see here, this is process that I have mentioned here. The first basically thing that we have to worry about is it is known as the recognition of the 5' splice site. As you can see here, I'm not going to be using the red pen. This is your 5' splice site. I hope you remember that. The U1 snRNP, this is U1 snRNP. This recognizes the 5' splice site. How does it do that? It uses the base pairing of, you can say it uses RNA-RNA interaction. Okay. What happens after that? Now, after that, basically, this thing comes U2AF65 and U2AF35. These are U2AF, which stands for U2 auxiliary factors. Okay, so U2AF, which consists of U2AF65 and U2AF35, they comes, and basically, let me tell you, the U2AF, as you can see, it is binding the PY tract, the polypyrimidine tract that we have. The U235, it binds on the 3' splice site. So I have labeled it here. Okay. Now, the U2AF, basically it also interacts with the BPP, right? I hope you remember the BPP, the branch point binding protein. And it is, as you can see, it recognizes the branch point, and it is, you can say, loaded here with the help of your U2AF. Okay. In the case of the mammals, the BPP is, instead of BPP, we have SF1. Okay, and this basically where the U1 is present on the 5' splice site, BBP is present on A or the branch point site, U2AF65 is recognizing the polypyrimidine tract, and U2AF35 is recognizing the 3' splice site. This forms the early complex, or you can say the E complex. Okay, now after that, I think I have already told you that this guy, the BBP, the branch point binding protein, will be replaced by U2, right? So U2 snRNP comes, it replaces this guy, and it takes the branch point, right? Now, the base pairing, there is obviously base pairing between the U2 snRNA and the branch site. And this base pairing, as you can see, it creates this bulge. I hope you can see it, because earlier when the BBP was binding here, there was no bulge, but as soon as the U2 binds, there is a bulge, there is a noticeable bulge here. Okay, so this thing where the U2 replaces the BBP, this forms the A complex. After that, what happens? As you can see, now we are going to replace. Okay, guys, I'm going to be very quick here, and I'm going to tell you the function of the U1, BBP. These guys, their function is basically identification, or you can say recognition of the main sites which are present, you can say in exon-intron junctions. Okay. After that, as you know, now after A and the B complex, we are moving toward the actual process. So as you know, the A complex is formed. After that, this guy, the U2AF65 and 35, these two brothers, what we're going to do, we will be replacing them. And who will be taking their place? This complex, which contains 3 snRNPs or snRNPs, U4, U5, and U6. And as you can see, they are now gone, and on their place, they are basically replaced by these guys. Now, a little bit information here about this thing. This is known as, let me tell you another name for this. This is also known as tri-SNRP particle. Okay, so that is just another name for this complex of snRNP U4, U5, and U6. Now, what happens here? The U4 and the U6, I hope you can see here. This is the U4, this guy, and this is the U6 on the side, right? They are held together by, you can say, strong complementary base pairing. Their snRNAs, they have a base pairing, so they are held together tightly. However, the U5, the guy in the back, is like just, you can say, a tag along. So the U5 is loosely associated here, okay, and After this thing where these two are replaced, this forms the B complex. Now, E means early. After that, we are just going A, B, and now C. Okay, so let's move them to C complex. Okay, so this is going to be a C complex. There are a number of steps that we need to take care of here. What happens that we're going to first replace this U1. The U1 is gone. It is replaced, as you can see here. Just take a close look here, right? The U6 and the U1 are present both in the close proximity of the 5' splicing site. Now, the U1 is here for identification purposes. Okay, he is not the main guy. He is just here to scout the target. The main, you can say, hitman, the U6, has now arrived. So what it will do, it will just say to U1, okay, buddy, I got it. So the U1 basically is replaced by U6, who now takes control of the 5' splice site. Okay, okay. So now the U4 again, as you can see now it is done. The U1 is replaced. After that, the U4 leaves. Right, the U4 leaves. And why is that? Okay, let's take a look. The U2 and the U6 are the main guys here. They need to work together because remember, this is the first trans esterification reaction. This bond will be broken and will attach to this point, forming a lariat, right? So in order to do that, the U6 must interact with U2, but there's a bond in the middle, the U4. So what we do, we just move it. The U4 is released now, and U6 can now directly interact with U2. After this, there is catalysis, right? The active sites of the U2 and U6 snRNAs, basically they prime the substrate for splicing. The active sites of these things, they you can say they juxtaposes. They basically, you can say, as you can see, there is this very high bend here that brings the 5' splice site and the branch point near to each other, or you can say together. Now, actually, again, I hope you remember how enzymes work. They don't actually do the job. They just make the conditions in which those reactions are favorable. So that's what they have done here. Because let's go back slightly. Here, as you can see, the branch point and the 5' splice site, there is a lot of distance between them. What's happening here, as you can see, uh, this thing, they have put these two things together so that reaction can happen. So this is their catalysis. After that, this is, as you can see, the bond breaks, and this is the first trans esterification reaction. Now, after that, what happens? You can say the second trans esterification reaction. Okay, so basically, the second trans esterification reaction, it occurs between, I hope you know, the 5' splice site and the 3' splice site that we have here, and it is done with the help of the U5 guy. Right? You see the guy in the back, the U5 snRNP. It brings the exons together. I know I cannot show you how it works, but it does, right? Because it is a 2D picture, and what I am trying to tell you is basically a 3D inspection, right? So just imagine that this guy grabs this thing and brings it closer to this one. However, in the 3D world, it is much easier. So after that, the second reaction takes place, and after that, you can say what happens? The exon product is released, the spliceosome disassembles, right? Now, this is how it works. However, you have to remember that this is not a foolproof reaction. Uh, Before moving forward, I would like to tell you something that the spliceosome assembly, right? It is, you can say, adaptable enough to ensure that splicing progresses in the cell. Basically, typically, it starts around the intron to be removed and can also initiate around an exon, right? But there is a specific process that we call exon definition or defining an exon, which is done with the help of splicing enhancer. So there is something else here that I'm just going to mention. It is not related to the splicing machinery. I'm just going to mention it here. The splicing enhancers are also a thing. They enhances the splicing process. Splicing enhancers, right? Now the sequence of event, right? I hope you remember these 7 different steps that I have mentioned here. The sequence, you might see that this sequence is there, right? But it is not fixed, guys. It is variable. The sequence might vary. This is, you can say, the most acceptable mechanism, right? But the sequence can vary, right? Furthermore, the spliceosomes ensures that the splicing reaction always proceed in the forward direction. You have to remember that as well. The splicing reaction that we have here, it always goes forward, never backward. And why is that? Let me tell you. Now, let's just say that the splicing process is always forward, so that it cannot go in the backward direction, right? So how do we ensure that? We can ensure that by, as soon as the splicing is done, right, we need to disassemble this thing, right? Because the splicing cannot happen by itself, at least here. However, in some cases it can, but here it cannot, right? So this thing, these guys are the one which are catalyzing the reaction. So what if, as soon as the reaction is done, we remove them, right? We disassemble them. And this thing is actually facilitated by splicing enhancer, another one, known as, let me just mention it, it is known as the PRP22. Okay, this is the PRP22, it is like, you can say it is a helicase, not a DNA helicase, it is more like a RNA helicase, right? Again, it is ATP dependent, as most helicases are, right? And it does a lot of things, right? I'm just going to mention them here quickly. For example, it is also involved in the second trans esterification reaction. So let's mention that, second trans esterification. So it is involved in that. With that, it is also, you can say, involved in the mRNA release. As soon as the exons are joined together, it ensures that they are gone, right? And it also, you can say, ensures that the spliceosome disassembles. So spliceosome disassembly, basically. So this thing is quite important, your PRP22. This is basically a dead box helicase, okay? Now, if somehow you can mutate the PRP22, the splicing can actually go in the reverse direction, right? So if the PRP22 is not there and the spliceosome doesn't disassemble, so it can go back in the backward direction. So technically, this reaction is reversible. But the PRP22, you can say, it ensures that it doesn't go in the backward direction, right? So now let's move forward. What's the next thing that we have here? Oh, as you can see here, we have the major and the minor spliceosome, and they have almost the same mechanism. However, there are some differences that we have here. Okay, I hope you can see it. On the one side, on the left, we have the major spliceosome. On the right, we have the minor spliceosome. Right? Now, the major spliceosome part, we are already done with that, right? The minor spliceosome, again, the complexes are the same. The E early complex, A, B, and C, right? But if you just pause this video and take a close look, you will see something else, right? For example, here we have U1, on this end you will have U11. Here we have U2, here you will have U12, right? Here we have U4 and U6. In this side, you have U4 ATAC and U6 ATAC. U5 is the common one between them. Okay. Now, most of the reaction again goes almost in the same way, right? But the thing is, almost everything else is almost the same. Okay. Just remember the differences, and U5 is common between them. Just remember that as well. So this is the major and the minor spliceosomes. Now let's talk about three different classes of RNA splicing. Now, basically there are three. For example, the first one is what we call the nuclear pre-mRNA splicing, as it is mentioned here. So then group 1 and group 2. You can mention them in any order. Okay. The group 1 and the group 2, these are self-splicing. So let's actually group them together. Let's make a circle and let's just write that they are self-splicing introns. And as the name suggests, they doesn't require the spliceosome because they can do it by themselves. They are self-splicing. Okay, so self-splicing basically means that the intron is kind enough to fold itself into a specific conformation, right? What conformation? This conformation, as you can see here. This is the intron, right? And here it needs the spliceosome to make this conformation. But in the self-splicing, the RNA or intron is good enough to take this conformation by itself. So as a result of that, it doesn't require any external help, and the splicing can happen by itself. Now there are some differences. For example, the group 1 and the group 2, they are quite rare, both of them, right? As you can see, their abundance, here we are talking about abundance, they are both very rare, right? On the other hand, the nuclear pre-mRNA, the common mechanism that we have just discussed, it is very common and it is used for most of the genes that we have. The group 2 and the group 1, they are rare and seen in some specific genes, okay? The mechanism is, again, as you there is one thing that I would like to tell you. In the group 1 introns, the branch point is a G. Just remember this thing, okay, that the branch point is a G in the group 1. In the group 2 and the pre-mRNA, the branch point is A, right? Mechanism is almost the same. There are some minor differences, right? I will be discussing that. I think I have the slide prepared. No, so I will be explaining that myself, right? So the mechanism is almost the same, but the group 1 has a different branch site. Okay, actually, you know what? Let's talk about them a little before moving any forward. The group 1 introns, which are actually self-splicing, they have a branch point G. Okay, so basically, everything else almost remains the same. Just the lariat is not formed. In the group 1 splicing, the group 1 introns are smaller in size. Okay, not as large as the pre-mRNA, the nuclear pre-mRNA. They are around 400 to 1000 nucleotides long, right? And they contain, I don't know, okay, fine. Actually, let's write here. We have the group 1 introns. The things about it, it is self-splicing. So let's write that down. It has a branch point G. So let's mention that the branch point is a G, not an A. Okay. Now, no lariat formation, no lariat formation, and almost the thing remains the same, right? For example, actually, I can make it myself. For example, imagine that this is your exon, right? The intron itself just makes a specific conformation here, allowing the self-splicing to happen, and then we have this exon 2. Let's call this exon 2. This one the exon 1. So there is a G present here, the guanine, and it has the same OH group. However, this is a 3' hydroxyl, not a 2'. It attacks the 5' splice site, the bond breaks, and the bond rejoins with the exon 2. This is quite easy. And basically, the introns, these guys, right here, they have a specific sequence. We call it the EIGS, also known as the internal guide sequence, which guides the intron to take a specific conformation. Okay, now again, this is a nucleophilic attack, guys. Everything else almost remains the same. The group 2 introns, these guys, the group 2 are actually, it is believed that the modern nuclear pre-mRNA splicing mechanism has evolved from the group 2 self-splicing. That's why, if you take a close look here, I have put them closer to each other because they are almost the same. The structure, again, there will be a lariat formation here, right? It is almost the same, guys. It is almost the same, right, as the pre-mRNA ones. However, there are some differences, but let's not go into that detail. Okay. Okay. After that, I think that is enough about your splicing. There are some things that I would like to discuss before moving any forward. There are two main issues with the splicing, right? So, it is very important for the cell to perform splicing on very specific locations, right? So, or you can say the splice sites. So for this to happen, the splice sites must be recognized by the spliceosome. The first thing that the spliceosome ensures is it should bind the right splicing site, right? The 5' splice site where the U1 and U6 binds, and these interactions are sequence specific. But we must consider the fact that some genes, for example, the dystrophin gene, are very large. The dystrophin gene is around 2400 kilobase pairs. and it contains a number of you can say very large number of exons. So it is possible that the spliceosome, or you can say the splicing machinery, can make some mistakes. There are two common mistakes that can be you can say that can happen. The first mistake is what we call the exon skipping. So there are two problems in splicing. The first one that we call it the exon skipping. So basically, as the name suggests, because the mRNAs, the pre-mRNAs, sometimes are way too large and contain a number of different splicing sites, that some exons might be skipped, right? So how it happens? Let's take a scenario. For example, actually, let's make a big. So imagine that this is a pre-mRNA, right? Okay. So and there are, let's take 3 exons. This is the exon number 1 that we have, and let's make another one right here. And then there is this one, right? So this is our exon. This is also an exon, and this is also an exon, right? So what we need, we don't want these introns, right? So I'm just shading them. We don't want them in the mature mRNA. So sometimes what can happen is that imagine that the splicing machinery picks up this exon using the splice sites, picks up this one as well, but doesn't recognize this exon. So this thing can result into what we call exon skipping. So basically what happens here, this exon, the guy in the middle exon, is skipped. As a result of that, the product which will only contain 2 exons, right? This guy and this guy, but the middle exon is skipped. This results into the most, you can say, the common error that we have. We call it the exon skipping. Now, there is another error that can also happen here. We call it the pseudo splice sites. As you know, the splice sites, they are recognized by their the splicing sites. They are recognized by their sequence. Sometimes the introns might contain what we call a pseudo splice site, or you can just call it a false splice site. Because if you remember, the conserved sequence is, for example, for the 5' splicing site, the conserved sequence is GU. So it is possible that sometimes, let's take another scenario here. Let's, for example, here we have 2 exons. Let's take, this is our exon 1, this is our exon 2. So let's write that down, exon 1 and exon 2. Here we have. Now, sometimes it is possible that the pseudo splice sites are present in the intron, or it is mostly common that the pseudo splice sites might, for example, imagine that there is a pseudo splice site here, right? So pseudo splice site. So now this kind of error happens when there is a pseudo splice site, and what will happen here? The spliceosome will recognize these 2 sites, the 5' and the 3', right? And instead of recognizing this site, right, which is your 3' splice site, what will happen? That instead, the splicing machinery will recognize the pseudo splice site. As a result of that, what will happen? It will contain the complete exon 1, so there will be no troubles there. So this is your exon 1, it is completely recognized. But for the exon 2, only half of it is recognized. So let's make, so as you can see, it will only recognize this part. So this is, you can say, the 2 most common errors that can occur during the splicing process. Now, the cell, because if this happens, the protein found will be completely useless. So, and as you know, the process of protein synthesis takes a lot of energy. We don't want that, right? So in order to make sure that these 2 errors doesn't happen, there are 2 methods, right? So there are 2 methods that are present to avoid these errors. So let's discuss what are they? The 2 methods to avoid these errors. Okay, the first one is what we call, let's just underline this one. Okay, so the first is what we call the live tracking, right? Live tracking. This is the first mechanism that the splicing machinery uses to prevent these 2 guys, the exon skipping and the pseudo splice sites. Now, as you know, during the process of transcription, the splicing proteins, these guys, the splicing proteins, they are present on the RNA polymerases CTD, right? So they are present where? the CTD of the RNA polymerase. So now you do also remember, I hope, from the previous video of the transcription, that the tail, the CTD of the RNA polymerase, is present quite near to the RNA exit channel, where the RNA, which has just been transcribed, you can say exits. So as soon as you can say just as soon as a splice site comes out, these splicing proteins which are present on the CTD, which is quite near to the RNA exit channel, they bind to it, right? So as soon as, for example, imagine that let's take another mRNA here. Okay, so imagine that this is a scenario and it contains 3 exons. So let's make 1, 2, and 3, right? So obviously this is the 5' end, this comes out early. So as soon as the first site arrives, this is your first site, the machinery will bind that. The splicing protein, it binds it. As soon as the second splicing site, which is the branch point, comes out, the next protein binds to it, right? And now it will obviously the RNA, the process of transcription goes from 5' to 3' the direction, and the 5' end is released first. So this is coming out in this direction. The RNA leaves the polymerase in this direction. So next site that will be coming out will be this one. So this is also recognized, right? Then comes this one, and it is also recognized. So using this live tracking method, the RNA polymerase, or basically the cell, ensures that none of the exons are skipped. So this is the one method that we have. However, there are some other methods. The second method that we have, it is known as the SR protein method. So let's write it down. So the second method is our SR protein. Now I hope the S and R are basically, we are using the denominations for the amino acid. S stands for serine and R stands for arginine. I hope you know that. So SR basically means that serine arginine rich proteins, right? So these are your SR proteins, serine arginine rich proteins, and they basically, they bind uh, to the specific you can say sequences that we have. We call them the exon splicing enhancers, the ESE. So where do these guys bind? They bind to ESEs, okay? And this stands for just your exonic splicing enhancers. So let's write that down. So they bind to that, and that basically it helps, right? It helps in what we call exon definition. Exon definition, again, let's take a let's make actually another pre-mRNA. So here it is another, I have taken another pre-mRNA for examples. It contains 2 exons, the exon 1 and exon 2. The exon 1 contains the sequences that we call the ESE, also known as the exonic splicing enhancer. So they are usually present, for example, like this. So these blue things that you see are the exonic splicing enhancer. They are present in the exons, right? And these are basically, you can say, these are the sequences where the SR protein binds. So let's take the SR protein. So this is your SR protein. The SR protein binds to these ESEs, and as a result of that, it helps in what we call the exon definition. Using the SR proteins, the splicing machinery, you can say, it is able to find out where the exons are and where is the intron. So as you can see now, because obviously your splicing machinery is blind, it cannot see, right? So it comes around here and then it finds this SR protein, and now it's know that okay, this is your exon intron junction and this is a 3' splice site, right? So it will recognize this and bind here. Then here the SR proteins, they help in the recognition that okay, this part is an exon. As soon as the exon ends, then you will have this one. This is what we call the 5' splice site, right? And so as soon as the SR protein is finished, the splicing machinery can recognize this splice site and attach here. Then it goes on, and there is no SR protein, so it doesn't know that. Now it here, as soon as the another exon comes, it recognizes this SR protein and it knows that okay, this must be the 3' splice site, right? So using this mechanism, using the SR proteins, the splicing machinery is able to recognize the exons, and this process is known as the exon definition, right, or defining the exons. So this thing can be used by the splicing machinery using the SR proteins, and it helps in, basically you can say, getting an idea where the exons are and where the introns are, so that the splicing machinery doesn't skip any, you can say, it doesn't skip any exon, and it doesn't recognize any pseudo splice site. For example, imagine that there is a pseudo splice site here, but as the splicing machinery is coming, there is an SR protein here which tells the splicing machinery, okay, that this is not a splice site, this is actually an exon. So that's how the splicing machinery uses your SR proteins to find out where the exon is and where the intron is. So these guys, these are all your SR proteins. So these are the two mechanisms that are used by the splicing machinery. Now let's talk about the variants of splicing, right? There are some variants that I would like to discuss here. So the next thing that we're going to do is we're going to discuss the variants of splicing. The first variant that I would like to discuss with you guys is what we call the trans splicing. So this is the first variant. The normal splicing that we have seen, right, we call it the cis splicing, right? When the both, just a second, I will explain them in a minute. So basically, for example, let's take another pre-mRNA. So just a second. So let's take another pre-mRNA. So this is your pre-mRNA. It contains, this is your exon 1, and let's take, this is your exon 2. So now the cis splicing is when the exons which are, which we have to join, the exons involved in the process of splicing are present on the same mRNA. So if I combine these two guys, right, that will result into what we call the cis splicing. Cis means on the same side, right? So the exons and both the exons 1 and 2 are present on the same RNA. So cis splicing. In the trans splicing, something unusual happens. What happens? So let's, for example, imagine that this is your exon 1, right? And as you can see, I'm using a different color. So this is your exon 1. And then let's take another one. This is your exon 2. Okay, so this is the exon 1 is present on RNA 1. Let's call it RNA 1. And the exon 2 is present on your RNA 2. Okay, so as you can see here, the exon 1 and exon 2, which we are going to join, are present on two different RNAs, RNA 1 and RNA 2. In that scenario, if the splicing happens, if you basically, if we are joining this exon with this one, this will result into what we refer as the trans splicing. So as you can see, this will be the result. So let's make just a moment. So this will be your result, right? So this kind of splicing is known as the trans splicing. And I will give you some examples. For example, it is most commonly seen in a nematode C. elegans, right? It is seen there. Most of its RNA actually, or the C. elegans, they undergo trans splicing. However, cis splicing can also be seen in the elegans, right? Now, the mechanism here, or the trans splicing, is almost similar to that of the cis splicing. Except there is one difference. There is no U1 snRP involved. Okay, because if you remember correctly, the U1, its main function is to recognize the 5' splice site, and if the U1 is, you can say, is present, let's just say U1 plus, which means U1 is present, it will result always in cis splicing. If the U1 is minus, or you can say absent, it will result into the formation of what we call the trans splicing. So these are basically, the U1 is responsible for that, right? These two scenarios can be seen. Now, another variant of the trans splicing, or the splicing that that is quite common, right? We call it the alternate splicing. So let's discuss that. The second one that we have, the alternate splicing. Now, the alternate splicing is, you can say, Okay, imagine that in what happens in the alternate splicing, the exons are deliberately, or you can say by, they are skipped. I know previously I told you that exon skipping is an error. Okay, it is an error when it happens by mistake, but sometimes the cell wants to skip exon, right? And that scenario is what we call your alternate splicing. Okay, let's see. Let's take a look here. I'm going to take an example for here for the troponin T gene. Okay, and actually, the alternate splicing, where the exon skipping is done deliberately, it is quite common actually. Let me give you a stat here. The 40% of the genes in case of Drosophila, they are made using the alternate splicing, right? And in the case of human, it is much more than that. Around 90% of our genes, right, are alternatively spliced. So that is the scene in the case of human beings. Now, let's in the case of human beings, I'm taking the example of the troponin T. In order to understand that, what I need is basically a pre-mRNA. So let's make one. So as you can see here, we have a pre-mRNA, and this is a pre-primary, you can say, RNA transcript of the troponin T gene, right? So there are, as you can see, the yellow ones are the exons. So name them. Let's 1, 2, 3, 4, and 5. So there are 5 exons here, 4 introns. Quite simple. Now, the troponin T gene, not only in humans, actually, it is quite common in the mammals. Now, the pre-mRNA contains 5 exons. But the thing is that the mature mRNA only contains 4. So what happens here? Okay, let's discuss that. What happens here? So there are two possible methods of, you can say, splicing here. So let's discuss them one by one. So if the mature mRNA contains 1, right, 2, 3, and 5. So the number of exons is 1, 2, 3, and 5. This will result into what we call the alpha troponin T. Okay, but in the other scenario, let's take a look. For example, let's just erase these things, right? And let's do it again. The another one is what we call the beta troponin T. So this is another, you can say, isoform protein, right? So it will contain 1, 2, 4, and 5. So as you can see, 1, 2, 4, 5. In the alpha troponin, what we have done, we have skipped the exon number 4. In the beta troponin T, what we have done is we have skipped the third exon, right? So this, using this process of alternate splicing, you can get different protein products from the same transcript or the same gene. However, you must remember that this process is highly regulated. It cannot happen just by its own. In the case of humans, I have given mammals, I have given you this example, the troponin T gene. For example, in the case of Drosophila, there is a protein. It might come in some exam. I'm going to tell you anyways. This known as the DASCAM gene. It is present in a Drosophila, and this gene is actually quite large, right? And containing a large number of exons, basically. And there are a number of, you can say, different variants that can be produced. However, this is again in the case of the humans. I have given you the example that we have used, the troponin T gene, right? So you can use that. Now, there are some other methods that you can also use, and actually they are quite easy to understand, guys. So for example, I'm going to give you a brief idea what are the other mechanisms, or can say what are the 5 ways, you can say 5 ways to splice a RNA, or you can say a pre-mRNA, right? So what are those 5 ways? We have discussed some of them. The first one is obviously the normal one, right? The normal splicing happens if all the exons are present and then we have removed all the introns. The scenario takes place. The normal condition will happen, right? Another one, for example, if you have skipped some exons, right? So that scenario will be what we call the exon skipped scenario. So this is the second possible way of splicing, right? Then we have something else. We call it the exon extended. Now you're going to ask me, sir, what is this exon extended? It is self-explanatory actually. For example, imagine that this one, it contains all of the 5 exons, right? Imagine that this is a normal one. But what happens that along with this exon 2, we also take some part of the intron. This scenario is known as the exon extended. Okay, where the exon will be present, but along with some exon, there will be some intron, some part of intron remaining. So this scenario will be known as your exon extended. There is one scenario as well where we call it the intron retained, right? And again, it is self-explanatory, in which all the exons will be present, but some part of the intron will be there as well. Not some part, one intron is retained, right? And then we have the one that we have just discussed, the alternate splicing. So these are the different ways that you can splice a pre-mRNA. I have mentioned them all here. I hope this doesn't need any further explanation. However, if you still want an explanation, just comment, just write it in the comments, and I will make a short video on that. Okay. Now let's come back to actually the troponin T gene, right? Now I told you that this process of exon skipping is highly regulated, right? The alpha troponin T and the beta troponin T, this process is highly regulated. Now the question arises that how does a cell ensures exclusive splicing? Okay, means how does it know that, okay, I have to take the third, I have to take the fourth, right? So there are three methods that can be used here. Let's take a look at those. So using these, you can say so using these three methods, uh, we are going to know how this happens. The first method is what we call the steric hindrance. Now, as again, it is self-explanatory. If the splice sites, as you can see, these two exons, the exon 3 and exon 4, are present in a close proximity of each other, the exons are too close together, right? So the issue is the two splicing sites again uh, are too close to each other. So the splicing factor, because I told you that the spliceosome is very large, it is almost as large as a ribosome. So it cannot just bind this site as well as this site because of the steric hindrance, right? So this again here what happens, sometimes either the exon 3 will be picked or the exon 4 will be picked. So this scenario is what happens due to the steric hindrance when the two exons are present together, right, almost together. So this scenario can happen. Now this you have to understand this occurs due to the relative positions of the splice sites. As you can see, this is the 5' splice site and this is the 3' splice site. They are so close together that it is too small to accommodate both the splicing machineries. One that recognizes 5' splice site and the other that recognizes 3' splice site. The second combination that I'm going to mention here is using different spliceosomes. As if you remember from the prior in this video, I told you that there are two spliceosomes, a major and a minor spliceosome, and they have slightly different splicing sites. So what we can do, we can use different spliceosome for different scenarios. For example, the major and the minor, their splicing sites are different. I hope you remember. Let me mention it anyways. For example, let's write it down here. For the major, the 5' splice site has a conserved sequence GU, and the 3' splice site has a conserved sequence GAG. So this was the scenario in the case of major. In the case of minor, what do we have here? Here we have like AU and AC. These are your splice sites, right? So as you can see, these are different. The splice sites are different. So what we can do, we can use minor for one exon and major for another one. So that method can also be used. It is actually the human genome contains a gene named, I think it is JNK1, right? And that uses this mechanism actually. Now, there is one more method. We call it the NMD, and it is somewhat different than the previous two. Now you're going to ask me, sir, what is this NMD? It is quite simple. It means that nonsense mediated decay. Okay, nonsense mediated decay. Okay, what happens in this nonsense mediated decay? This mechanism actually does not force splicing to be mutually exclusive, but it ensures that only mRNA, you can say only mRNA with either one exon or the other survives. So imagine that in this scenario, both of these are formed, right, by the same major spliceosome, because sometimes it will recognize this site, sometimes this will that spliceosome will recognize exon 4. So the thing is, sometimes you will get 50%, you will get this one, 50% you will get this one. But nonsense mediated decay means that the desirable product. So imagine that the cell only needs this one. So this mRNA, right, uh, will be targeted for degradation. Okay, so it doesn't force to pick this or this. What it does, it will allow both of them to be formed in their proportions, right? For example, here 50%, 50%, and then it will degrade the incorrect splicing product. As simple as that, right? So that can happen. Now, the alternate splicing, I'm telling you again, it is highly regulated. And in the regulation of this, something comes into play. What is that? I have already mentioned the SR proteins. The SR proteins are also used here, right? So the proteins that regulate the binding to the specific sites, we call them the splicing enhancers and the silencers. So let's mention them as well in the alternate splicing. How is it regulated? Right? Actually, I'm going to mention it right here. In the regulation part of the alternate splicing, we have two things, right? We have the splicing enhancers. These are also known as the activators, so you can use any term. And the second one is what we call the splicing silencers. So these will be acting as, you can say, these will stop, right? So enhancer enhances, the silencer suppresses or represses the nearby splice site. So using this as well, the SR protein, the serine arginine rich protein, it will bind a particular exon. Okay, if it is, you can say enhancer, it will enhance the splicing of that particular exon. If the silencer binds to that exon, then the splicing of that exon will be enhanced. For example, in this case, let's take another example. Actually, let's make different shapes here. For enhancer, I'm going to use this shape because it is in the upward direction, so it enhances. And for this one, I'm just going to use a simple rectangle. Okay. So imagine in this one, we want beta troponin T, which will include exon 1, 2, 4, and 5. So what the cell will do, it will bind a silencer on the exon 3, which will silence it, right? It will not allow it to splice, and then bind an enhancer on exon 4. Now, using these two enhancer and silencers, the cell increases the splicing of the exon 4 and suppresses the splicing of exon 3. As a result of that, only the beta troponin T will be formed. So this can happen, right? So this is, you can say, method how using which it can the cell can regulate the type of splicing that it wants, right? So these are basically your, you can say, the variants of splicing. I'm only going to discuss two. However, there are more, as I have already mentioned here, but we don't need to go in that detail. I'm going to give you some examples here, obviously, for splicing enhancers and the regulators. Okay, for example, in the case of the Drosophila, I told you there is a gene named DASCAM. If you remember, it is mentioned on the left top corner, right? So the silencer for that scenario is going to be your HRP36. So this is an example. I'm just going to write DASCAM so that you guys can remember. It is in the case of the Drosophila, right? Then let's take another one just to show. There is this snRNPa1, right? This is another silencer. It is it basically, I think it targets the HIV TAT3 mRNA. But again, you don't need to go into that depth, right? So these are two repressors. And actually, there is one more, hnRNP1, also known as a PTP protein. Your PTP protein, as mentioned here, it is, you can say, it binds to the exon, right? And it basically, you can say, inhibits its splicing. right? It blocks the interaction between the exon and the protein, for example, the U1 snRNP, right? So this can happen. Now, I'm going to give you some more ideas here that the alternate splicing, you can use it for a number of different manners. For example, the alternate splicing is actually used in pluripotency. But again, if you want, I think the name of the topic is alternate splicing and pluripotency. So basically, it is related to your embryonic stem cells or the ESCs, right? But I'm not going to mention it here because again, it is a way deep topic. But if you want a separate video on this, I think it will be a 5-minute topic, not a big one. So if you want that, you can just write in the comments, and I will make that one too. Okay. So basically, this is how it works. So now we are done with the variants of splicing, the errors that can happen in the splicing, and how the cell avoids those errors. So this is all about your splicing. So I forgot to mention one more thing that we need to discuss actually. The thing is that I hope you know that the introns are only present in the eukaryotes. So they are not present in the prokaryotes, right? So the issue is, the question arises that how does the exons came into play, right? How does the exons evolved? So we're going to discuss that here. So basically there are two models, right? Uh, and again, as you know, that the prokaryotes doesn't contain any introns. So there are two models that can explain the origin of introns. The one is known as the intron early model, right? And the second one is known as the intron late model. So I will be giving you a brief introduction to both of them, and then you can decide. Again, these are both hypotheses, right? You can decide which one you like. So the intron early model, again, it is self-explanatory. That intron, it says that introns existed in all organisms, including the prokaryotes, but they were lost from the bacteria during evolution. Because again, uh, just let's write this down first, that the introns, the first point here is that the introns existed in all organisms, including prokaryotes, right? But they were lost from the bacteria. From the bacteria or just from bacteria during the process of evolution, right? So this is your intron early model. So the theory behind is that is because again, you know that the bacteria is the simplest organism possible, right? It doesn't contain any kind of organelle, it doesn't have a nucleus. So in order to the introns existed in the bacteria, but in order to make things more streamlined, they just they deleted it basically. As a result of that, you will always see that the bacterial genome is always very small as compared to the eukaryotic genome, right? So the thing is, the intron early model suggests that they were present in almost all organisms, but prokaryotes, bacteria, deleted them during evolution, right? So, and as a result of that, now their genome doesn't contain any kind of introns, right? So this is your intron early model, right? Now there is another model, and I hope you can guess the name of it. It is intron late, right? So you either arrive a party early or you arrive the party late, right? So this is the intron late model, and again it is quite self-explanatory. It says that the introns never existed in bacteria. During introns, you can say, appeared in eukaryotes during evolution. Appeared in eukaryotes during evolution, and they never existed in the prokaryotes. Okay, so now again, which one is right, the intron early model or the intron late model? So uh, the thing is, doesn't matter which one is correct, you can pick according to your choice. I mostly prefer the intron late model. I will give you my reasoning, and if you like the intron early model, I would like to hear your reasoning in the comments. The intron late model implies that they were never present in the prokaryotes. During the evolution, as the organism became more complex and complex, the introns started to appear because they were quite useful, right? You can do exon skipping, exon extension, alternate splicing. There are so many significance of them. And again, earlier in this video, I showed you a graph which shows you that the number of introns increases with the complexity of the organism, right? So using that, you can say that okay, even the small eukaryotes, for example, Leishmania major, it has some genes which doesn't contain any introns. Yes, it has some introns, but some of them doesn't exist. So it states, but as you move forward in the evolutionary ladder, you come to the Drosophila. It contains some introns, not many. Then you go to the mammals, the rat and the human. They contain almost 8 introns on an average. So it states that with evolution, as the organisms became more and more complex, the number of introns kept on increasing because it was a useful thing. So eukaryotes evolved with it. Right? Again, intron early model has also has their benefit if you go with the intron early model. But the thing is, doesn't matter which one you like, the intron early model or the intron late model. There is a question that I would like to ask you: why the introns were retained in eukaryotes? Why introns were retained by eukaryotes? Right? Retained in eukaryotes, because obviously at some time evolution, the bacteria has been on this planet for a very long time. They must have developed some kind of intron, but they didn't keep it, but it was kept by the eukaryotes. Why is that? Now uh, see, there is a clear advantage. I told you that there is a clear advantage here, that the presence of intron, right? And there is a clear disadvantage as well. Let's talk about the disadvantage. uh, if there is intron present between the exons, you have to remove them and you have to spend a lot of energy on that. So that is a disadvantage, right? But there is an advantage. If there are introns, you can do exon skipping, you can do alternate splicing, right? You can do exon extension, and all of them are useful, right? You can reshuffle your exons, that you can also do. You can do cis splicing, you can do trans splicing. So there are so many benefits, and that's why I think the eukaryotes retain them during evolution, because eukaryotes are complex organisms, so they needed some complex you can say complex mechanisms. So my answer to this question is modern problems require modern solutions. So that is why the eukaryotes retain them. Okay. Now, according to me, basically there are three main arguments to support this view. Actually, right? Uh, the introns were beneficial for the eukaryotes. There are three main arguments that we can use to support this view. Let's see what are they. The first one, that according to me, I would say that the first argument which supports this theory is that the border of the exon and intron, right? The border of exon and intron is usually it lies at the boundaries of protein domains. At the protein domain, I hope you know that proteins domain. A protein can have multiple domains. So usually one exon makes one domain. The next exon makes the next domain. And using this method, with each exon, they encode an independently folding unit of the protein and often corresponds to an independent function. So technically, that's how it works. For example, imagine that there is a protein which has an ATPase activity as well as some DNase activity. So you will always find that the ATPase domain is different. For example, let's make a protein. So it is something like this. So it will have an ATPase domain and then it will have a DNase domain, right? So as you can see, they have two different domains, both of them. They fold independently, and secondly, they perform their function independently as well. However, they are connected, right? Now, another supporting evidence that we can use here, that it is possible that the introns, right, many genes, not introns actually, that the many genes in the proteins that are encoded by the exons might have evolved using exon duplication, right? So exon duplication might have been a reason behind that, or maybe divergence, right? So for instance, imagine that a protein is composed of repeating units. For example, let's take the immunoglobulins, the antibodies, right? So I hope you know that the immunoglobulins, they contain repeating units. So it is possible that at first there was only one exon, But due to exon duplication, there was another exon just like that, and that actually is the reason how immunoglobulins or the proteins which contain repeated units might have got those repeated units. So that is another thing that you can use, right? There is third one. For example, let me again, I'm going to take an example here, before explaining it to you. For example, the LDL, right? I hope you know what is an LDL. LDL stands for low density lipoprotein, right? The low LDL, basically the LDL receptor gene, right, which makes the LDL receptor, it contains exons which are evolutionarily related to something very different, which is the EGF, the epidermal growth factor precursors, right? So this suggests that due to the divergence of the exons, right, due to that the exons present in the EGF precursors are also found in the LDL receptor. That is my argument. So what does it suggest? That it suggests that the exons present in the EGF precursor proteins, right, they somehow got, you can say, transferred or duplicated into the LDL receptor. So that is my third argument, that related exons, right, related exons are sometimes found on completely different proteins, on completely different proteins. So again, using these three arguments, I explained why eukaryotes retained the introns, right? They were useful to them and they evolved them and retained them. Okay. Among these, the intron early or the intron late model, I leave that up to you, which one you prefer.
Thank you for watching. I hope you found this video helpful and informative. If you enjoyed the content, please give it a thumbs up and share it with your friends. Don't forget to subscribe for more videos. Hit the bell icon to stay updated with our latest uploads.